So, Guus, the floor is to you. Thank you for being with us today, and we're looking forward to your talk.
Right, thank you, Manon, for a very nice introduction. Can you see my slides?
Yes, thank you. If you put it in presentation mode, we're really happy.
How does this look?
Not quite there yet. But maybe if, yes, that's it. Thank you.
All right. Okay, thanks again for a nice introduction. So I will be talking about some research that we have done in our lab regarding the development of more universal influenza vaccines. Well, in order to understand the immunology and epidemiology of influenza viruses, I first will introduce some general properties of these viruses in the next couple of slides. So influenza viruses are viruses with an envelope, and in this envelope we can find a number of membrane glycoproteins like the hemagglutinin and neuraminidase. And these are important proteins because they're not only involved in the virus replication cycle, they also target for especially humoral immunity and protective immunity. So there are 4 types of influenza, A, B, C, and D, but only A and B are clinically relevant for humans. And within the type A, several subtypes can be distinguished, which are also defined by the hemagglutinin and neuraminidase molecules. Furthermore, it's important to note that the genome of these viruses is a segmented genome, which means that quite easily in double infections, genetic information can be exchanged by reassortment events. And that's also important for flu. So, as Manon said, these viruses are highly contagious. They're airborne, it's airborne transmission. And yeah, as a result, we can see epidemics almost every year. The clinical impact has been mentioned by Manon already. But why are these viruses so successful in causing outbreaks almost every year? And the main reason for this is that these viruses can change, right? So we call it antigenic drift, which is caused by the accumulation of mutations around the receptor binding site of the hemagglutinin, which allow these viruses to evade recognition by neutralizing antibodies that have been induced by previous infections in previous years. And as a result, we see, like here on the right, every year we see this emergence of clades of new variants that are antigenically distant from their ancestors. And as a result, the vaccines also need to be updated almost annually to make sure that the vaccine strains match the epidemic strains antigenically. So this is a complication. Furthermore, I've talked about seasonal epidemics caused by antigenic drift variants, but we also know that influenza viruses can cause pandemics, which are worldwide outbreaks of influenza. Most of the times, these viruses are reassortant viruses, having gene segments obtained from human influenza viruses, but then they have obtained a new subtype of hemagglutinin and neuraminidase molecules to which the population at large has not been exposed to. And yeah, then these viruses can cause worldwide outbreaks of influenza. So we have epidemics and pandemics. And then since 1997, we also know that, that these viruses occasionally may jump from animal species to humans, like zoonotic infections. And there are many subtypes that have been shown to be transmissible from animals to humans, and also causing severe disease. And a few famous ones are, of course, the H5N1, which also started in 1997. And also H7N9 has been famous, causing severe disease in human subjects. So when a pandemic strikes, the clinical impact is enormous. There's always excess morbidity and mortality in the population. And these are the most recent pandemics, as we know. The last one was in 2009. This was called Mexican flu or swine flu. Also causing hundreds of thousands of deaths. So I think we all agree that, these viruses cause a threat It's not if, but when the new pandemic will come. There's no doubt about it that we will see new pandemics in the future, but it's very hard to predict which subtypes will cause the next pandemic. And even if we have a suspicion which subtypes can cause a pandemic, then it's very hard to predict which variant of this subtype will cause a pandemic. For example, H5 has been considered a big concern. It's also posing a pandemic threat. And the WHO Global Influenza Surveillance and Response System is generating vaccine strains of all possible different clades of H5N1 viruses. But still, we cannot predict which of these clades will possibly cause the next pandemic. So that is a problem. The other problem is, to make a vaccine in the face of an emerging pandemic, it may take too long to produce tailor-made vaccines. It may be quicker nowadays with the advent of the mRNA technology, but still it takes a long time to make tailor-made vaccines. And it is recognized that the availability of vaccines that can induce more broadly protective immune responses would be helpful to mitigate the impact the next pandemic may have. So if you want to make a universal vaccine, by definition, you need proteins that are highly conserved, right? So you need antibody or T-cell responses directed against proteins or fragments of these proteins that are highly conserved. There's been much interest in the induction of cross-reactive antibodies, and especially the stalk region of the hemagglutinin is an interesting target for the induction of cross-protective antibody responses. And it has been already demonstrated in animal models that this can be achieved by hyperimmunization or by passive transfer of antibodies directed against this region of the hemagglutinin molecule. You can get a broadly protective response. Well, as Manon indicated, I have an interest in T cells. Also, T cells are interesting because the majority of CD8 positive T cell responses are directed against the structural proteins like the nucleoprotein and the matrix 1 protein, the internal structural proteins, which are way more conserved than the hemagglutinin and the raminidase. And also the polymerase proteins are relatively conserved. So this could be an interesting target for induction of cross-protective T cell responses. Well, as a matter of fact, we know this for a long, long time. So this has been demonstrated also in animal models, but more than 40 years ago, Andrew McMichael and his colleagues already showed in a chimp study, so in a human infection model, that the extent of lytic activity afforded by CD8 positive T cells inversely correlated with the extent of viral shedding in volunteers that were experimentally infected with an H1N1 virus, to which these subjects had no antibodies. So indicating that indeed the CD8 positive T cell responses are an independent cord of protection against virus replication. And as I mentioned, the majority of these T cells, the CD8 positive T cell responses, are directed against the internal proteins like the MP and the M1 protein. Well, in 2009, we had an experiment of nature. So we had a pandemic caused by an H1N1 influenza virus. And a group in London from Streetheart et al. Showed that people, it was a prospective study in which they showed that people with a high frequency of CD8 positive T cells directed against conserved epitopes less likely developed severe disease with fever. So also in this case, the magnitude of pre-existing T cell responses correlated with less severe illness in these patients. And then also for the zoonotic viruses, it has been demonstrated that people that have experienced a infection with zoonotic virus of the, for example, the H7N9 subtype, that recovery from these infections is also correlated with a very dominant and robust CD8 positive T cell response, that is shown here from a group in Australia. So we have an interest in using this, right, in exploiting CD8 positive T cell responses in a way to make vaccines that may be able to contribute to more broadly protective immunity. We have been involved in a European project, so we have been coordinating this project. It's a European-Indian partnership, so we have also colleagues in India working on this. So we are looking at ways to induce cross-reactive antibody responses. But here in red, I will be talking about today is the construction of an MVA polyepitope construct for the induction of cross-reactive T cell responses. So what did we do? So we designed a synthetic gene consisting of ubiquitin, because it will dock a synthetic protein to the proteasome for optimal proteolytic degradation of this protein. And the protein consists of conserved epitopes, CD8 positive epitopes, separated by spacer sequences to optimize clean cleavage of the epitopes by the proteasome. So, and then of course, if you want to make a vaccine that's just based off epitopes, you have to consider also the HLA coverage, because each of the epitopes will be presented by a single HLA allele. So we selected 20 epitopes. We started looking at the immunoepitope database. We found 972 epitopes, but then we down selected. So we only looked for epitopes in the polymerase proteins and P and M1. We only looked for a certain size, and we also wanted to make sure that the antigenicity of these epitopes was already confirmed by others, right? So they, if you use these epitopes, they already induce interferon gamma responses. And then this is, of course, very important. We wanted to make sure that we selected highly conserved epitopes. And in the end, we selected 20 of those, which an HLA coverage of over 99.5%. So that this potential vaccine could be used in a wider population. So this synthetic gene was then cloned into a viral vector. It's called MVA. It's a modified vaccinia virus ankara. And we use this viral vector because it's highly attenuated and replication efficient. And it's also known for its capacity to induce protective humoral and T cell responses. Of course, the synthetic gene can be delivered by other ways, for example, by mRNA, that would be possible as well. But we had access to this technology and we used it to make the proof of principle. Of course, we had to characterize the construct, so we confirmed the insertion of the gene of interest. We showed expression of the protein, and we also confirmed this virus was still replication deficient. So, having made this construct, we first wanted to make sure that it was antigenic in vitro. And the way we approached this was by using PBMCs that we stimulated with peptides. So we generate peptide-specific T cell lines, and then use these T cells to cultivate with either HLA-matched transgenic A549 cells. So we have this in the lab. We can use those as antigen-presenting cells, or autologous monocytes. And indeed, we were able to show that the peptide-specific T cell lines could recognize and could be activated by antigen-presenting cells that were infected with the MVA polyepitope construct. We show this here for a number of different epitopes. And also, if you use professional antigen-presenting cells like monocytes, we also see that the polyepitope construct can activate those peptide-specific T cells. And then we did the same for T cells that were not expanded by a peptide, but by influenza virus stimulation. And basically, you get the same result. So these are influenza virus expanded T cells that then were activated by antigen-presenting cells infected with the polyepitope construct. also did the other way around. So we called it in vitro immunogenicity studies, where we stimulated PBMCs with the polyepitope construct, an empty vector control, or influenza virus as a positive control. And then after 12 days or so, we isolate the T cells and we activate those with antigen-presenting cells that were loaded with the peptides. And as you can see that with the polyepitope construct, we get nice responses that are very similar to the ones that we observed after influenza virus stimulation. So the polyepitope construct was immunogenic in vitro, just like the influenza virus. And even the immunodominance hierarchies between different epitopes was very similar between the polyepitope construct and influenza virus. So yeah, we did that for a number of different epitopes. So we showed for 3 different epitopes and 2 donors each that indeed the polyepitope MVA construct was immunogenic in vitro using PBMCs of HLA-typed blood donors. Next, we wanted to confirm that the construct was also immunogenic in vivo. And for that, we used HLA transgenic mice. So these mice are transgenic for HLA-0201. And the mice were immunized twice with a 3-week interval. And then 2 weeks after the last immunization, the mice were sacrificed and the splenocytes were tested for the presence of influenza virus-specific T cells. And we stimulate those splenocytes either with peptides, which are then positive controls, the polyepitope constructs, or the wild type. And the readout was interferon gamma production by interferon gamma 80 spot or ICS. So here we see the results. So if you look here on the right, this is a re-stimulation of the splenocytes with a peptide that is derived from MVA. You may appreciate that both the recombinant MVA polyepitope construct and the wild type empty vector both induce T cell responses against this peptide. Whereas the response to an influenza-derived peptide, in this case from the matrix-1 protein, we only observe that in the mice that receive the polyepitope construct. So from these experiments, we concluded that the polyepitope construct is immunogenic in vivo and induces T cell responses to MVA and influenza-derived peptides. And we also analyzed the T cell response by flow cytometry, which confirmed that the responses that were induced by the polyepitope construct were indeed CD8 positive T cells. So the H5N1 viruses that currently cause so much problems in the United States and elsewhere also attracted our interest, and we were interested to see to what extent our polyepitope construct could, potentially protect against infection with these H5N1 viruses. So we compared the sequences of the epitopes with those present in the influenza H5N1 clade 2.3/4.4b strain. As a reference strain, we used a Kettle, Texas 2024. And it turned out that 17 of the 20 epitopes were fully conserved in this virus. So 3 had a difference, 1 amino acid difference. And of one of these 3 epitopes, we were able to check whether this mutation from a tyrosine here to histidine had an impact on the recognition of virus-specific T cells. That is shown here. So here we did again an antigenicity test. So we expanded T cells by peptide stimulation using the wild type or the H5N1 derived peptide. And yeah, here, as we have seen before, you get a nice response with the polyepitope construct and a very potent response to the homologous peptide, which is much lower for the variant H5N1 variant. But expansion with the variant peptide was actually not so successful. You may appreciate that the scales are completely different here. So this response was way, way lower. So we already concluded that there's limited cross-reactivity with the variant MP44 epitope. And then we also did the immunogenicity experiment again. So we stimulated PBMCs with the polyepitope construct and checked if the expanded T cells could recognize the homologous peptide, which it did, as expected. But the variant epitope derived from the H5N1 virus was not recognized, indicating that this mutation is detrimental for recognition of MP44 specific T cells. And most likely this is due to a decreased binding of the epitope to its corresponding MHC class 1 molecule, because the ninth position of this dimer is actually the anchor residue for binding to MHC to HLA A201. Oh, no, sorry, HLA 0101. So this is too bad, but in the polyepitope construct, we have multiple A1 restricted epitopes present. So to conclude this, we designed a synthetic gene consisting of 20 highly conserved CD8 positive T cell epitopes. These epitopes were fused to ubiquitin and separated spacer sequences to ensure optimal antigen processing and presentation. And we demonstrated antigenicity of this construct using different antigen presenting cells. We demonstrated immunogenicity in vitro using PBMC of HLA type blood donors, and also in vivo using HLA transgenic mice. We found a patent application, and we also produce a seed batch that is ready for production under GLP conditions for testing in clinical trials. So finally, I want to acknowledge especially Shrumista in our lab, who has done most of the work on our side, and Alina Czerne at LMU in Munich, who did most of the work on their side. And of course, I have to acknowledge funding, European Commission for funding the ENFLU project. And with that, I want to end. Thank you very much for your attention. And if there are any questions, I would be happy to address those.
Thank you, Guus. That is really exciting work, and it does raise a couple of questions from my end. But I want to invite the audience also to type their questions in the chat box, and then I will, revert them to Guus. But Guus, I would like to start with the following. I was a bit surprised that you didn't select any epitopes from the stem of HA. Is there any particular reason for that?
Yeah, so there are two reasons. So the HA stem, of course, is more target for antibodies. What has been demonstrated, antibodies against this region are, exert protective effects. Okay. And we did not select any epitopes from the hemagglutinin because after natural infections, CD8 positive T cells against the hemagglutinin are hardly induced. So it's very, very rare if you look for HA-specific CD8 positive T cells. So they're not induced after natural infection with influenza. So the vast majority of T cells are directed against the internal structural proteins like the nucleoprotein and the M1 protein and the polymerases.
Okay, thank you very much for that. And then I was also wondering, would you expect a T cell vaccine to be effective in elderly people? As we know that, the T cell immunity really, goes down as you grow older? Yeah,
so that's an interesting question. So one thing we have to consider is that T cell immunity alone will not afford sterile immunity, right? So as a standalone vaccine, this will not fly. It has to be an adjunct to existing vaccines to bolster T cell immunity against influenza viruses. And I think that also elderly people may benefit from that if you can bolster their T cell responses against influenza.
Okay. And then actually, final question from me before I go to the question from the audience. would you consider, I mean, especially since you work at the University of Veterinary Medicine, to just test this vaccine in cattle first? Because it seems to be so much easier to do challenge studies in cows than just going to humans, right?
Yeah, but then the biggest problem is HLA restriction, right? Or MHC restriction. So the epitopes that we have selected will not be presented by MHC molecules.
So it doesn't work.
It doesn't work. In cows, it doesn't work. It only will work in humans.
And there's not enough knowledge to translate this to, you know, a cow vaccine.
To cows, no. I mean, I'm not a cow immunologist, but you know that will be interesting to select epitopes that are actually presented by cow MHC molecules. But I'm not sure how well that has been investigated. I'm not sure.
All right, so let me go to some of the questions from the audience. Is there concern about anti-MVA responses blunting transgene-specific CTL responses on future immunization using this vector?
Well, that's a very interesting question that we actually have addressed some years ago. So we have looked at the immune responses induced by a recombinant MVA expressing hemagglutinin in mice that were already pre-exposed to either anti-vector, or we even used the pox vaccine that has been used to protect against smallpox to see if the pre-existing immunity against this pox viral vector had an effect on the immunogenicity of the recombinant. Okay. And that effect was very, very limited. So even in the presence of pox viral immunity, these recombinant MVAs are still immunogenic. And I don't know why that exactly is, but these are very big viruses. So maybe it's not so easy to neutralize these viruses upon repeated immunizations.
Okay, thank you for that.
And Dr. Cox, Dr. Peter Palese has had his hand raised for quite a while, so I was going to allow him to ask his question directly.
Okay, can he do that?
Yes. You can go ahead, Dr. Palese.
Yeah, I don't see anyone raising his hands. That must be in your,
I think he is on mute.
Okay.
Can you hear me now?
Yes.
Yes.
Okay, great. Thank you. This was a very elegant presentation, Guus, and congratulations. I have one question. Is there any quantitative number which you think a T cell vaccine, even if you, as you said, you want to have it in addition to vaccines right now, but is there any number of T cell of a titer which you think would be helpful and protective?
Yeah, that's a good question, Peter. So, I mean, the minimal requirements for protection are unknown. I mean, I have to be honest with that. Just looking at frequencies, I think is not enough, because it also depends on the quality of the response and the epitope that is recognized. Right? There are some low affinity, high affinity receptors. Their epitopes are inducing much better and stronger T-cell responses than others. So it's not just the frequency, it's also the quality of the T-cell response that plays a role. So I think it will be a big challenge to define minimum correlates of protection for these T-cell responses. That will be next step. You know, this is really a difficult one to crack.
I think maybe related to that, thank you, Peter, for that question, is a question from, I think it's Han van der Bos, asking whether you did any animal challenge studies with various flu types.
Yeah, so we plan to do that, but then in the end,
to do that?
Yeah, because the number of mice that we could obtain for these studies was too limited. Yeah. So we had to refrain from the challenge experiments, but it's still something that we still would like to do.
All right, well, Guus, I would like to thank you very much. In the interest of time, there are quite a few more questions in the Q&A box. It would be wonderful if you could take a look at those and type your answers. And thank you to the audience for your questions.
Well, thank you for the opportunity. I'm going to sort of give a run through of what we've done over the last 4.5 years to bring FluMist for self and caregiver administration to the U.S. market. It really provides an innovative approach to help prevent influenza. And because this presentation, I'm giving it from the US, and it is an approved drug. There are some approved vaccine. There are some things that I will have to mention, including some important safety information and the indication. So we really had, during the pandemic, there were some really important things that happened that drove the interest and ability to file a supplemental NDA for FluMist to be used by self or administered by an adult to an eligible child in the home setting. There was really a rise in healthcare consumerism. We got used to, and the US population got used to having everything delivered. I mean, everything from toothbrushes to pet care. And that really extended into the healthcare realm, where we see now the availability of testing for respiratory viruses, RSV, not yet, but for flu and COVID. We see Cologuard being available and being sent directly to the home. And then we also see injectables being available. And this really gave the opportunity to have an interest and the feasibility of having self-administration and caregiver administration for FluMist in the home setting. And this really provided an opportunity to do some additional research that was required by FDA to do a supplemental BLA for FluMist in the home setting. I do need to touch on the indication for FluMist, and I'll go back and forth between FluMist and live attenuated influenza vaccine, which is the generic. In the United States, and I know this probably has an international audience, in the United States, FluMist is approved for persons 2 to 49 years of age for the prevention of flu, and it's administered intranasally. There are other indications elsewhere in the world. FluMist is an egg-based vaccine, so if you have a severe allergic reaction or anaphylaxis to any component of the vaccine, particularly eggs, or to a previous dose of flu vaccine, it's contraindicated and for children and adolescents receiving aspirin or aspirin-containing therapy, it's contraindicated as well. So FluMist for self and caregiver administration that can be given in the home or essentially outside of the traditional healthcare delivery system reduces barriers to care and helps improve, is thought that it could help to improve vaccination rates. And patients will benefit from this is the same recommended and approved FluMist vaccine simply delivered to the home. It really extends who can administer the vaccine. So traditionally, the vaccine has been available in the United States since 2003 and could be administered by healthcare providers. And this really changes who can administer. So an adult 18 and older can administer it as a caregiver to themselves or to an eligible 2 to 17-year-old. How it can be done is that it's really, and I think I have, if we have the time, the ability to show you how easy it is, but it can be given in the home setting using an established online pharmacy service. So this is still a prescription medication. It's not over the counter, and the screening processes remain the same, but it's done through an online pharmacy service. And it really expands access and empowers individuals to administer the vaccine really not only according to label, but also with the previously established ACIP guidance. Again, the original approval of the product was in 2003, and we've demonstrated effectiveness comparable with injectable flu vaccines in the US and globally. And there's really quite a bit of safety data available in the PI as well as in the global literature. I do want to touch on vaccine effectiveness, as this question comes up quite a lot, and this is the last few years of vaccine effectiveness. Some of this data has been published in a recent concise reports. If we just look at this year's data, the 24-25, most of this is interim data. This purple color is live attenuated influenza vaccine or FluMist, and This is injectable vaccine, and you can see vaccine effectiveness is comparable. We do have recent data from the US at 60-point estimate of 61% compared to 48%. This is data out of California, and the rest of this is out of the EU and the UK. And again, you can see it's really quite comparable over the last 3 years. And actually, this data would be consistent if you look at even over the last 20 years. We just published some of that data. So this data, along with safety data, should give folks confidence that FluMist given by self at home or take by caregivers will provide comparable effectiveness. There is a small series of publications that were used to submit as part of the supplemental BLA. Some of these are a little bit older, but they do all demonstrate that individuals can safely and effectively administer the product to themselves or to others. The initial clinical study, the labeling clinical study that was done in the 1997-98, it's hard to believe I'm talking about something so old, but in the 97-98 original study, there were 4,500 or so individuals, and a majority of those folks they were given the choice to self-administer or to have administration by a healthcare provider. And a majority of those folks administered to self and did so without difficulty. Close to 100%, it was 96%, felt that they could do it without difficulty. And immune response in this study, effectiveness and side effects were similar whether the individuals administered to self or a healthcare provider administered it. Burgess, which is part of a Department of Defense study, again, demonstrated that immunogenicity and side effect profile were similar, whether given by self. And again, most preferred to administer to self. A small study by Ravi Javari sent product home with families, and there was a strong preference for families to administer at home. And many in this study said that they would not have had access or been able to get flu vaccine for their children had they not done it through this manner. And these other two, this study really was interesting. It was a pandemic planning sort of exercise where they had small groups of individuals who self-administered under the direction of a healthcare provider. And there was a small series in the UK actually where they actively vaccinate children in school, and they were able to have children self-administer, which we won't be recommending in the US. And they were actually able to provide direction to children to self-administer, but there was a lot of poking and sort of having fun. And while the children were able to do it, it was really not a very efficient process, and they use nurses to administer in that setting. I do want to touch on the safety profile. It is this is a topically administered or nasally administered vaccine, and it does differ than injectable vaccines. So with this vaccine, you see side effects at the site of administration, which is the nose. So we typically see a runny nose and congestion. And when you have a runny nose and congestion and you can't taste anything, you tend to see a decreased appetite. And we see that with FluMist compared to placebo, and FluMist compared to an active control, which was injectable vaccine. You can see the side effect profile is very consistent. And when we went from the trivalent to the quadrivalent and back to the trivalent, the side effect profile has been remarkably consistent over 22 years in the United States. You can see a fever with FluMist. We've not seen febrile seizures. Important to note, we typically see a fever with the first dose of flu vaccine received. And in these studies, which were done in the late 90s and early 2000s, this was often the first vaccine that children received. In the United States, most children by age 2, which is when they would become eligible for FluMist, have received at least a single flu vaccine. For adults, again, we see a runny nose and headache and a sore throat. And that runny nose, usually for both the adult and the pediatric population, we typically see about 48 hours after administration of the dose. And that's because that's when you sort of see the peak of the attenuated virus replication. And so it's essentially driving that immune response. With the approved for self and caregiver administration, there is a single package insert, whether it's delivered, the product is given in the clinic or their home. There's not two package inserts that go to different audiences. It really is just an extension of who can administer the product. So it's always been available for healthcare providers, and this really extends that availability of who can give it to adults and caregivers, and that's based on eligibility. And there's more information now in the package insert on how to administer the product and a little bit more on side effects and allergies. It also really includes very specific information regarding self-administration and administration to individuals 2 to 17 years of age. And it really specifies that children should not self-administer the product and that caregivers or parents should administer, someone over 18 should administer to a child, an eligible child 2 to 18 years of age. It has a unique, there is a unique packaging for the self-administration application, and I'll show you that in a minute. It has a unique drug code, NDC code, that's specific for the single dose carton that's used for billing. There's also a unique CDC CVX code that is used for documenting in immunization information systems in the United States. Importantly, there was some regulatory items that we really had to address in order to submit to the FDA. And that was to demonstrate that the product could be used safely and effectively without any concerns of harm by individuals. And when I say harm, I mean whether they could fully administer a dose and not mist it into the air or just give it in one nostril. So this is done through a process called human factors engineering. And for those of us who have lived in infectious disease for most of our careers, or even primary care, we're really not that familiar with the whole concept of human factors engineering. If we were in pulmonary medicine and we're using things like aerosolized products or inhalers, all of those products go through a human factors engineering to ensure that the product can be administered in the device effectively by the patient. In this setting, it's a iterative process where you have to look at everything from the labeling, the packaging, the artwork, where the package insert is, where the directions are, and to ensure that intended individuals can effectively access and use the product effectively. And we see sometimes common mistakes where the package is opened incorrectly, and you even have to modify the packaging to ensure that the package is opened correctly. And I'll again show you what that looks like. We choose a patient population that looks like intended users, so right and left-handed persons, different genders, different age groups, the wide range of education and literacy backgrounds. And the final study that was done demonstrated that 100% of intended users were able to safely self-administer the product or could effectively administer it to an eligible child. And that sort of information is submitted to FDA as an important part of the approvability package. So upon approval, which was just this time last year, really provided the opportunity for FluMist to be administered at home. And it provides an additional option for flu vaccination, really increasing access and availability of flu vaccine, and provides the opportunity to increase vaccination rates. So the usability tested, we feel really confident that adults will be able to effectively use the product based on our human factors usability testing. This is what the single carton looks like. It has this sort of artwork. It really clearly shows you what the product will look like. And once the box is lifted, and there is really a very specific way where it says lift here, and the box opens without ripping to reveal this. And this is one of those things that had to be changed through human factors, where people open the box incorrectly and would dislodge the vaccine. And so this actually was one of the first things we had to change, was even how the box was labeled with the lift open here. It seems like a little thing, but did make a difference in folks being able to use it. So there's a really quick note here that says a full dose equals 2 sprays, 1 in each nose, in each nostril. And then it goes on to have additional instructions that I'll go through in a minute. So this is what the initial instructions look like. 1 dose equal 2 sprays, 1 in each of the nostrils, and really does highlight the importance of the divider clip, which I'll show you in a minute. Upon opening the package and prior to administration, there are some really clear instructions on how to use the product. So it should suggest that you have a clean surface to work on, to check the expiration date. These are all things that are obviously already done by the pharmacy dispensing the product, but there's a request that the patient check it again. So check the expiration date. Don't use the product if it's been damaged or tampered with. And then once you open the package, so if I go back here, when you remove the instructions for use, the vaccine lays right there, and you essentially are only able to pull it out in one way. It's sort of a natural placement of the fingers, so that you pull it out by the barrel of the device and not by the plunger, and then provides instructions on how to inspect it. And then steps 5 through 12 really are how to administer the product, to pull off the gray cap. Don't remove the divider clip. We say this many times. And to put the vaccine into the nostril, it doesn't have to go all the way into the nostril. It just needs to be just inside the nose. And then to depress the plunger quickly, and not to stop until you hit the dividing clip. And only at that point does it tell you to remove the dividing clip, and then repeat steps 9 through 12, which is the continuation of the vaccination. There's also a couple really good notes in here that speak to the fact that the most common questions we get, which are sometimes you see a little dripping from the nose, and that's normal, and we have that even in the instructions, because it's a very common event. And then the step 13 speaks to the disposal of the product and the program that's in place to return the sprayer so it can be disposed of properly as medical waste. And I'll get to that in a minute. Now, in order to get the product to patients, it goes through an online pharmacy service. And the online pharmacy service fully supports the patient population interested in administering FluMist at home. And the program is called FluMist Home. So people tend to confuse this, but there's FluMist for self-administration, which is the indication, and FluMist Home is the service that supports FluMist for self-administration or caregiver administration in the home setting. So it's really an experience for those folks in the United States that is very similar to preparing for a retail pharmacy vaccine visit, where you enter patient information, you provide and do the eligibility questions that you would ask for either in the primary care setting or would likely answer online. You share insurance information. You provide a small amount of payment for shipping, which is $8.99 in the US. We anticipate, and what we've seen is that most insurance plans will cover this. And then you choose the date that you want the product delivered to the house, and then the order is submitted. And this just gives another little insight. Is that if you've done any sort of go into a pharmacy for a vaccine, you would be familiar with this sort of your basic information. And once you get the basic adult information in here, there's the opportunity to provide request product for other folks in your home, either another adult and your children. And for each of those, the screening process is included, as well as critical information like the patient's date of birth and allergies. This is a sort of a sampling of the medical questionnaire. It's really aligned with the ACIP guidance and derived from the immunize.org website. There are a few places where you can see where it would limit a person's eligibility to receive FluMist at home. So these are either ACIP guidances or contraindications for asthma and wheeze. And obviously, if you're egg allergic, taking salicylates, and so forth, you wouldn't be eligible to receive product at home, and you receive instructions to seek your vaccine through your healthcare provider. There is then, these are reviewed by pharmacists and other healthcare professionals to ensure that the eligibility criteria have been met. Individuals are given sort of the, green, yellow, red sort of symbols for whether someone is eligible, whether a person requires additional review, or the healthcare provider has questions. And then this patient is ineligible based on their age. We expect in the US that commercial insurance will pay for the product with very little out-of-pocket. So the out-of-pocket really is the payment for shipping. And FluMist will continue to be available in clinics and pharmacies and for VFC when administered in a VFC provider office. And we continue to work with and we'll talk with the VFC program office to see if how FluMist can be incorporated into that program. And again, here you're able to put in the date that you'd like it delivered to your house. There's no requirement to be present or sign for the packaging. And just like anything else that you receive and order online, there are extensive communications with order confirmation, status, including text message and emails to let you know if something's going to be delayed or not. The signature, again, is not required. And again, in the text messaging, you're also given other ways to find instructions for use, including videos on how to administer. I just want to point out the cold chain shipping is really important and something that we worked very closely with FDA to ensure we had a validated cold chain. So it's shipped under really strict cold chain procedures. And this was also part of the BLA. It requires 2-day or less shipping, and each package can hold up to 4 FluMist devices. It has instructions for use and a disposal envelope, the cooler, chill packs, an inner box that has the product, and it all goes into this outer box. This is all called a pack out. And upon opening the box, there is a temp tag where individuals be able to use their phone to determine if the product has stayed within the appropriate temperature during shipping. Upon delivery, this would be unpacked, and this package would go into the refrigerator for storage until the family was ready to use it. Once used, you'll receive a text. Once the product is received in the home, you'll receive text messages to confirm administration. If the patient does not respond, they'll continue to receive reminders. And if they don't respond, they'll be asked if there's a reason why and whether or not they need additional support from my pharmacist to administer the product. In the US, it's really important that vaccination is documented, and the process is the same as the retail pharmacy setting. And so once there's affirmation from the patient that vaccine has been administered, then the pharmacy, online pharmacy, will post that to the state-specific immunization information system. And individuals can download their vaccination record. And if they provide their physician's information in advance, it will be sent to their physician, and they can always provide that information as a proof of vaccination to their provider that they can put in their health record. And then finally, I just want to touch on disposal. This is the FluMist device is considered medical waste, and there's a return shipment program that's outlined, and this comes in the pack out. And this the used device or devices go into this little baggie, and then shipped in this envelope that can go right into the mailbox to a waste disposal company. So I'm not going to go through all that important safety information, but generally the product is not recommended for use in persons who are immunocompromised. It's not been well studied in that population, or in folks who have a history of significant underlying health conditions that may give them increased risk for undesirable outcomes associated with flu vaccine. And FluMist, like all other flu vaccines, may not protect everyone receiving the vaccine. And as I said, children who have a history of wheeze or asthma, the ACIP don't recommend use in. I'm not going to go through that. And so just finally, for what's happening this year is that the launch has occurred. It's available currently in the United States. Again, there's one package insert, and the product remains available in pharmacies, in clinics, and through the FluMist home service, so that the persons can administer to self or to caregivers, or a caregiver can administer to a 17-year-old who's eligible. And we're continuing to work on additional payment options, creating materials in other languages. Most of the materials right now, beyond the package insert, are which is available also in Spanish. Most of the materials in the online information is all in English, and we'll be looking into doing a final phase 4 effectiveness study in the self-administered population sometime in the near future. I think that's it.
Well, thank you, Allyn. This is really, really different from the scientific talks we had before, but amazing the amount of work that is involved in getting something done like this. So one of the questions was, of course, when will FluMist be available? You explained that it was not over the counter, that it's still basically managed by an online pharmacy. But have you already been receiving orders? Because not many people are aware of it, I guess.
I'm sorry, I missed that last.
Not many people are yet aware that you can just go online and order the product and get it shipped to home. So my question was, have there already been orders coming in?
So we launched formally launched in August, and the website opened, and it remains available. And I'm on the medical team, so I really can't speak to where they are with ordering, but I believe that there is an enormous amount of interest and uptake.
I can imagine. So can't wait to get updates on that. So we have a question from Karina Paskiewicz, if I pronounce this well. You mentioned that the vaccine cannot be used if you received a previous flu vaccine dose. How much time should you wait?
No, you can. Most children by the age 2 have received a previous flu vaccine, like over their first 2 years. And that was really around the side effect profile of a fever. So if with your first flu vaccine, children tend to develop a fever. With FluMist by age in the U.S. by age 2, most children in the U.S. have received a flu vaccine, so we don't see a significant fever in those children.
Right.
But there's for an annual vaccine, there would be no reason not to use it if they had received a vaccine, the year before. So I'm
Right.
not quite sure what she's asking.
So Allyn, there are a lot of questions in the Q&A, and I would like to ask you to take a look at that
I
and
will.
answer them while
Mhm.
we go on to the next talk, just in the interest of time, because
Yep, absolutely.
right, we are a little later. Thank you very much,